Hello, everyone. Thank you for joining us with this installment of Boyce Thompson Institute's Breaking Ground discussion series. We're glad you could join us today. My name is AJ Bushy, and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome Murli Manohar, a BTI alum who is now co-founder and chief technical officer at Ascribe Bioscience, which is a startup company developing natural compounds as crop protectants. And today we'll be talking about these compounds and what is next for the future of Ascribe Bioscience. So hi, Murli. Thanks for joining us.
Thanks for having me, AJ.
And okay, let's go up and away. Murli, can you please tell us a little bit about yourself, how you get into plant science and how you came to BTI?
Yeah, thanks again for having me, AJ. It's a wonderful opportunity to talk about what I have been doing at Boyce Thompson and eventually what transcribed into Ascribe Bioscience. But like you said, before we go there, let me introduce myself. So I'm from a farmer family. I was born and brought up in a state, it's eastern part of the country in India called Bihar. It's it's bordering with Nepal, so next country there. And so like I said, my father, grandfather, they were all farmer, and most of the crop, as if I remember correctly, was either potato or onion, some rice, and some vegetables. So, so you see the inherent inclination towards agriculture from the family already. And when I was young enough to help them, I usually go to the field and help them. And there were years when they get a lot of pathogen, and potato Phytophthora infestans is one of the major pathogens, and we get that in India as well. And the damage can be very severe. So that's where I got insight on agriculture and the problem farmers faces. And eventually, I did my schooling and early college in that state. And then I moved to another state, which is not too far from Bihar, called UP. That's where I did my bachelor's in agriculture, because I wanted to learn more about agriculture and, new techniques as it goes. That was 4 years program. After that, while doing that, I got very interested in the molecular aspect of the agriculture, the molecular biology and biotechnology. So I got into one of the IIT in India. It's called Indian Institute of Technology. That was a campus in very close to Himalayas, actually, called IIT Roorkee. That's where I did my master's in biotechnology, and that's where I got exposure to all genetics, genetic engineering, biochemistry, both human and plants. But my always interest in plants, so I learned both humans and plants. But eventually, after that, I applied to quite a few colleges in US because I wanted to pursue a PhD in plant science. And I came to Texas A&M University, College Station. I worked with researcher Professor Kendall Hersey. He's a biochemist. He works on nutritional genomics. So my project mostly was on biochemistry, understand the dynamics of how transporters move nutrients around the plants, and the focus was calcium. I also spent some time crystallizing and getting the three-dimensional structure of that protein. And while I was there at Texas A&M, we invited Professor Dan Classy, who was a researcher at that time. He was one of the guests for one of the seminar we organized there. And that's when I had a chance to interact with him. And I was pretty impressed with what he does. And when I graduated, I sent him an email, hey, Dan, if you're interested, I would love to come to your lab. He didn't respond for some time, so I thought he's not interested. But then he said, oh, I was in a conference. I saw your email, but I couldn't respond because I was traveling. And then he emailed me back, and then we had conversation. He said, I don't have position, but I think, you will fit in my lab. So that's why I got into Boyce Thompson Institute, started with Professor Classic. And then we had,
so what year was that or so, Murli?
Oh, good question. That's late either 2012 or early 2013. I think it's late 2012. So working with Dan, and then we had the lab had ongoing collaboration with Frank Soder. So that's my exposure to both lab, and eventually the whole story about the scribes came from this collaboration. So that's a little bit about me. Long story short, came from a small town in India.
Yeah, it's interesting how many people here in the building come from farming backgrounds, and and go into plant science to try to make it easier to farm, because farming is so hard.
It's hard in India. It's different in India. When I came in, I saw farming here. I think the farmers have much better equipment, better tools than the small state where we grew up, mostly manual labor based, and that's when you have more trouble and things can be very expensive. So it's different here, much better, but still, I think they all face some similar sort of challenges across the board.
So so let's get into Ascribe. It's a pretty interesting story how it came about. Could you tell us that story?
Yeah, and this story has been told many times, many ways, so I'll try to be as accurate as possible. So this started when Frank was out with his last coffee, 4 in the morning or 5 in the morning, between 4 or 5. And Dan Classic, Professor Classic, who comes early, so he was out for his first coffee. And that's when I think Frank was telling him the story about how he discovered a molecule, pheromone molecule from C. elegans. When when treated, when they used to treat mouse, they suppress the immune system. So Dan was immediately interested, asking back, well, is these molecule also present in the nematodes that's found in soil? And Frank was like, yeah, I think so. Or maybe he already knew back then. So that's the conversation. And Dan was like, oh, this is interesting. If it could be a common phenomenon across the kingdoms. So next morning, I think I just joined the lab, maybe a month in or so. I was working with a senior researcher, Patricia Manusalva. He's a professor now at UC Riverside. So we were both sitting in the office, 8 in the morning, having coffee, and then Dan walked in with telling us that this could be very interesting. And then project was assigned to both of us. And the first thing we took the molecule, Frank gave us some of the molecule, and we put it in the test against our favorite plant, which is Arabidopsis. And we found that quite opposite what it does in animals, it actually activates the immune response in plants. And so that's how we discovered, and initial finding was in Arabidopsis. Then we expanded to multiple crops. We had access to tomato and potato in the lab, so we could do those experiments. And then eventually we collaborated with multiple research outside to validate this in more in the commercial crops like the wheat, corn, and soybean eventually.
So I think one of the interesting things about these compounds is they really are like protectants, almost like a vaccine, and it primes the immune system in a way. Is that an accurate way to think of it?
That's right. So and there's a relation to be drawn here. So the several product that's out there in agriculture that's SAR based. So you call systemic acquired resistance. When you treat plants, it activates the immune response. So this compound also falls in that category, but it's a little unique in the category because it does not immediately activate the immune response. It basically just prime them. So so like you said, it's more like a vaccine to the plants. You go to pharmacy, you take the vaccine, and you come home, and you don't feel anything until the pathogen comes, and that's when your body responds faster. So it's unique in a way. We still put in the category of systemic acquired resistant based product, but it doesn't activate directly. It primes the plant. So plant can go by healthy without having any impact on their normal physiology. But if pathogen comes, they respond faster. And quite contrary, for example, a very good common product out there, Actigar, which is salicylic acid derivative-based product. You put that on plants, it's a red alarm for plants. They shift their physiological response to defense response mode. And now, if you do that, you are compromising normal growth and development. So our product doesn't do that in a manner that's pretty unique. And I don't know if any product out there that is similar to this.
So it sounds like these other products, they make the plant expend more energy on the immune system, keep them going and going and going and going, and then they don't grow as well. They don't have as high a yield. Is that what you're saying? Whereas
basically, it's in the scientific community, it's called growth defense trade-off. So if you are activating one part of the plant, then you are taking energy from other part of the plants, which is normal physiology. So it's going to impact the normal plant growth and eventually the yield.
And that doesn't happen so much with the Phytalix.
It doesn't happen, nothing. Plant goes, we haven't seen any negative impact on the plants after treating this. So we don't see any negative impact on the plants.
And before we get into more of the science, how has the company done so far?
Yeah, well, so there's a good slide right here. First, I think the phase 1 was really, and we put this together just to give everybody an idea of how far we had to come to start a company, and then how many more years we have to go to actually commercialize this technology. So starting from 2013, the first few years we were just testing this, validating this technology, right? You do in the model crops like Arabidopsis and the pathogen you in the lab. And then I think once we had confidence that this is actually worth, immediate value of this product was clear for agriculture. So then we get into starting the company, and that's when Jay Farmer, who is our CEO, he comes with 20 plus years experience in the startup company. He and I and with Frank and Dan started this company in 2017. And 2017 to 2019, we're just trying to figure out what to do. And I think that's the time when we thought the small business innovation research grants coming from federal agencies like NSF, NIH. We crafted those plans and we submitted some grant. And our first grant was NSF, SBIR phase 1. That was in 2019. So that's when we took this to pilot scale. So early on, you see how the things work in lab. You can infiltrate, you can put in the soil, but those are not viable approach when you do try to commercialize. That's when either you do seed treatment or you do spray treatment. So that's what we did with the phase 1. We just tried to test how viable commercially it is. And then with the spray treatment or seed treatment, it worked fine. Then we took it to the field. I think our first field trial was 2019, late 2019. And that's when we got the hint that this actually not just the lab event, it works in the field. And to give you perspective here, AJ, I think 95% of the product that's developed in the lab doesn't work in the field. So that was something keeping us all up at night, right? Because yeah, this all works in the lab, but what happens when it goes to the field? So that was a big relief in 2019. And then we came this far from one field trial. So now we have hundreds of trials done and more needed to be done for actually have a commercial viable product in the next few years.
And I think we have a map of where all those field trials are.
Yeah, and this is a little bit old map. This is from last year, but the overall structure has not changed. We have trials, all the regions you listed here, but more so than we had last year. We have 100 plus trials sponsored by Ascribe, and then we have similar number of trials sponsored by our partners. And last 2 years, we were mostly focused on wheat and soybean. And as you imagine, the small company, we can take over everything. So we strategically made decision to just focus on wheat and soybean. That's a big market by itself. 45 million acres in US for wheat and about 80-90 million acres of soybean. So it's a big market by itself. The last year, we got some hint that it also works in the corn, one of the major crop in US. And then also in rice. So this year trial, we added plenty of corn and rice. And the focus, all these crops, we chose the pathogen that's most relevant for farmers. And so we have pathogen ranges from bacteria, virus, less so on virus, but bacteria, fungi, oomycete. These has been major focus, and I think these, the fungi are appear to be a major problem in the row crops. So,
go ahead.
No, I was just finishing my thought. Go on, AJ.
I was just going to ask: is there any reason or that you might think of why Phytalix could work better in some plants than others, or against some types of diseases than others? Like you mentioned fungi, or is it just that's just the biggest problem, so that's what you're focusing on, or does it work better against them than other types of pathogens?
I think focus was row crops, and fungi is the major problem compared to any other pathogen. So that was obvious choice to choose if you have choose where you want to focus your resources. But that being said, we have tested against the virus and bacteria, and we have seen similar response, good response as well. I think some of the trials this year that's also focused on out there in the bacteria. And sometimes you are in the field, although the focus is fungi, but we get to see everything, right? Mm-hmm. So the field in South Dakota, for example, the focus if Fusarium has had blight, that's major focus there. But then we also ask farmers or contractors to look for any other pathogen comes. And what we have been seeing is you see the protection against all pathogens, and that's why we call this a product of broad spectrum. So sometime what happen is if you go in the field and you see not much disease there, maybe 5% here, 5% there. But if you combine 10 disease of 5%, then at the end they will drag the yield to significant number. And we have seen that even though it doesn't look like the plants are doing well and nobody cares for the 5% disease incidence or severity, 10 of those can make a big difference. So Phytalix, this is more like a preventative technology. So if you have applied that, and we have seen in some trials, you apply that, then you have protection, significant protection against all this disease, and then eventually at the end, you will have a better yield. So it's a broad spectrum. And also that being said, and two of the pathogens that we have not seen much of an impact, so I don't want to give an impression that this is a silver bullet. So the couple of pathogens, and the reason, likely reason, we don't know for sure, is because this technology works by activating defense response. If some pathogens are extremely aggressive, the plant may never have chance to activate their defense response. So we are trying to understand better, but we do have a couple of pathogens where the initial trials have not seen much effect.
Gotcha. And we have a neat little video, I believe, that will show how, one example, how good it is.
And what you're seeing is untreated plot. It's all dead. It's heavily infected with powdery mildew. So nothing left. It's all dead, no pumpkins here. Now, if we move, this one here is Phytalix by itself. And that is looking great, nice and green and good protection against the powdery mildew.
That's amazing.
I must say, AJ, that's the protection is not always this dramatic. this one was done by so our field trial manager, Tom Hedden. He manages our field trials, and his side kick or his hobby is to grow pumpkin. And he has this pumpkin, and he put on a trailer outside his farm, and there's a box you can put $5 and take one pumpkin away. So I saw that last year. I was like, hey Tom, can you just test in your system? We are interested in a specific crop, and this would be helpful. And he was like, yeah, it's no big deal. We can put some plot out there. And he carved out 4 plots for untreated and 4 plots for Phytalix treated. And I was there just a month ago, and I was like, oh my God, this looks good. This is so, I couldn't resist making the video because that was so dramatic there. But we see the trend in other crops as well. I have made video in wheat, corn, and soybean, and we see trend there as well. You can see the decent protection against the target pathogens, but clearly not as dramatic as this one. So I hope that everything looks as dramatic as this one, but that's not that's not what it is.
And it doesn't take very much of this stuff either, does it? This is tiny amounts.
Oh, it's very, very little. So just to put that in perspective, aspirin tablet, assuming that 400 milligram, that would treat 20 acres of wheat plants. And there's a variability in the optimal concentration between crop to crop. So that's wheat. And if you go to the rice, that needs tenfold less than wheat. It's you're dealing with 200 acres. And just to put that in perspective, the best-in-class fungicide, which is Prosaro, that uses 100 gram and 100 gram of tebuconazole and prothioconazole. So 25 milligram versus 200 gram. So you can do the math. It's already 1,000-fold more than what we are using.
Wow. So you said the size of an aspirin?
It's enough for 20 acres.
Wow. So
it's very potent, right? I mean, and it goes back to the basic of science, right? I mean, this how the plants respond to this molecule produced by nematodes in the soil, right? They produce this in I don't know, it's a really low nano or femtomolar concentration. So plants have adapted or evolved to sense this in the faintest amount present in the soil. So basically, actually, that does make sense. And there is an optimal concentration. And you were talking about this bell curve. So there is optimal concentration. It's not like you go 10-fold or 20-fold higher. It would still work. How the plant respond to this external signal is by coordinating through the receptor system on the surface. And those receptor system doesn't work if you have concentration out of normal, out of optimal. So I think this varies from crop to crop, but mostly in the range of, I would say, most of the crop, 25 milligram per acre, that covers most of the row crops.
So you bring up an interesting point there that we didn't really talk about earlier, was that the plants their roots are exposed to these nematodes, the C. elegans, all the time, and they've evolved to detect a teeny tiny amount. Then they know that these predators are going to come start eating their roots, right?
More or less, and that's their sign to signal for them to activate their defense response. And if you think about the plant defense response, it's not like us that we produced antibody for specific pathogen. Plant is, if you cover their major pathway, which is salicylic acid and jasmonic acid pathway, you are basically covered for most of the pathogen. Salicylic acid more so for bacterial pathogens, and then jasmonic acid is more covered to fungal, oomycete, necrotroph type pathogens.
Mm-hmm.
And it does target both of those and some other, the biochemical pathway. So it's target those and prime those pathways and provide protection against this multiple pathogens.
And it's also neat that it's a protectant and it doesn't actually kill anything. So I would imagine that's
a very I think unique distinction, right? I mean, all the fungicide out there, they are kill pathogens, right? You want to apply when the pathogen is present and you kill it. And that's all the problem with this fungicide, and that's major reason why the farming is moving in more sustainable way than using the old fungicides. These are toxic. The old fungicides are toxic. Some of them extremely toxic. That's if you go back and look in the Europe, they're removing these fungicides from the rate faster than US, and they are not environmental friendly. The rest stays there for quite some time. It goes into the water. So all sorts of problem. And I think one of the major problem is if you keep hitting the pathogen with these fungicides, the pathogens eventually gonna grow over it. They're gonna gain the resistance, right? So when they gain the resistance, these fungicides are no longer useful. So you need modern technology. And based on my conversation with some of the researchers, there's emergence of pathogens are getting faster and faster. I mean, time take to emerge a pathogen was, let's say back then, 20 years ago was 15, 20 years. Now it's 7, 8, 9 years because they're getting stronger and stronger. So farmers do believe that the biological or natural product is the way forward, but they're also worried that at this stage, the way it's sold to them is like, oh, this is going to change your farming, going to give you 20% yield boost. They also see that as a snake oil type of the product. So it's a lot of learning for them and a lot of I think all these large agrochemical companies reaching out to them and make sure they understand that this is actually a viable tool for the future. But on the same time, they also have to be careful not to exaggerate the benefit. Just give them in their hand and see, just look for yourself. And that's how we do, Ascribe does. We reached out to farmers and I was like, I think this is good. Here is the results from all this other experiment. I think it's going to work. Why don't you test yourself? And some of them has tested themselves, and they have said that this is actually really good.
So it sounds like sustainability is only really a selling point if it also works as well as other stuff and gives you that yield.
To get through the door, you have to actually prove that this actually works. And if it works, then I think the farmers are interested in biological. They understand the benefit of it. But I think if it doesn't work, which is the major problem with all the biologicals out there, is it's just inconsistency, right? It works this year, not next year. And the costs, the most of the biological product out there is only used in organic farming, which is what, 2 or 3% of the total farming, because it's too expensive. Normal row crop farmers can't afford it. Then it's all microbe based. There's a supply chain problem with this. You have to store in a certain temperature. You have to use in a given time, and all those problems that's the biological. And Phytalix has none of those. It is stable for years. You need so little. It provides consistent results across the board. It's environmental friendly. You're not putting some bug in your soil that may change the soil physiology for years to come.
Right.
So I think using clear, defined, natural molecule over a microbe, I think it's and the farmer gets it. But I think we still have a long way to go to convince that this is the better than all the other products out there.
And what is this graph we're seeing here now? What is a Prosaro?
Every time we do these trials, AJ, the farmers want to see how this compete with the current one. If current one is $20 per acre, and you're saying that yours is going to also cost $20 per acre, how does it compete with that? Why should I change this? So every trial we do, we got to bring this the best in class that they are using now. So what you're seeing here is Prosaro is a common fungicide used for Fusarium head blight, a major pathogen in wheat. So what we did, this is a trial was done in Maryland with one of the researchers there. And we asked the question, can we compete with the best in class? And what you're seeing here in the middle, that's the Phytalix, disease protection by Phytalix and disease protection by Prosaro. So I shouldn't say the Phytalix outperformed Prosaro, but it was very comparable to Prosaro. So that gives us hope that we can go out and compete with best in class. Although we have some more work to do in certain circumstances, like if you have a lot of disease pressure, the product that works through the plant immune system may not be sufficient. So what can you do? What other thing you can mix with so you have a viable product that's provide protection under modest disease pressure versus even the disease gets at a very high level. So something that's we are still considering and working on it.
Sounds like you still have a lot of field trials going on, and we'll have a lot of data coming out, I'm guessing, pretty soon.
Yeah, I think we have. We're getting some data now from all these trials. I think we have couple hundred trials across mostly in US. We have some in Brazil. We had some in Canada. We are expanding trials to Asian country. We are hoping to get some in India, Vietnam, Indonesia. So these countries, and you want to test in a different agroclimatic region. And some cases, the pathogen here is a major challenge, may not be the problem in Asia. So if you're developing a product for Asian country, you want to make sure that the product that also works in the pathogen system that's more relevant for the other countries. So we do have a lot of data coming in, and perhaps we'll have a better idea in a couple of months where we stand and what we need to do more next year before we launch in a few years.
And it's a little after 12:30. I invite our attendees out there to put some questions in the chat if you have any questions, and I'll ask one last one. What other projects are you working on over there at Ascribe? So
other than this agricultural product, that's we've spoken to, we are also expanding this to food safety. So if you think about the fruits and vegetables that you eat raw, right? It's coming from field, and there is multiple ways it can get infected by the human pathogens.
Mhm.
The contaminated water, animal feces, animal droppings, or equipments or human contact, all those things. And this food comes, and say you go to Wegmans and you buy this fruit platter. Those fruits are not treated with any product to kill the infection coming from the field. So one of the target is, can we use this product to treat the plants near harvest, and can they provide protection over the period after the plants are harvested, right? So then you have the chances of this human pathogens can be reduced that in the food from farm to fork. So that's like a somewhat post-harvest aspect of food safety. We are also very interested in the sprout. It's a different market. It's a fragmented market. It's not as big as post-harvest and an agricultural market. But I think we have all seen sprouts in the Wegmans or grocery store you go. It's a full of nutrition. There's a lot of proteins there. But then if you think about how the sprouts are grown, it's a nice, warm, and humid condition. That's perfect for microbes. Yeah. And seeds that's coming from the farm, most often they have a low load of these human pathogens, Salmonella, Listeria, E. coli, but the growth condition in the industry, that's really perfect for these microbes to grow log scale, and sometimes it gets pretty stronger. So we asked, if we are activating plant defense system, can we also provide plant protection against these human pathogens that is inside the seed, or when you grow inside the seedlings or microgreens. And there are current products that basically are like bleach based or peroxide based. These seeds are treated with so you can kill the microbes on the surface, but these microbes are not always on the surface, they're inside. So the only way to get to those is if your plant can fight themselves. So we are just trying to understand this better, and make sure and see if treating seeds can give the activated immune response to provide protection against these pathogens. And early results from our collaborators, Jinru Chen in University of Georgia, and she has shown that so you have negative control, that's basically uninfected in seeds, and then you have mock treated, which gets to 8 log fold after 7 days. And if you treat with Phytalix, nothing.
Wow.
So this is one of the fabulous results we got. And once you see these kind of results, you want to go back and repeat, right? This has been repeated multiple times, under multiple conditions. So it does provide a quite good protection. We have a little long way to go to scale up for the more commercial setting, but the results so far is looking good. So yeah, post-harvest, sprouts, microgreens, we are in that area as well. Yeah. And NIH was very interested, so we submitted this grant, 2 grants actually. One was focused on sprouts, so we got the phase 1, and this is the results we got in phase 1. We submitted the phase 2, so we got the phase 2 totaling about 3.3 million. And then we got another grant from NIH, which is most focused on post-harvest. That's that project is just starting, so we're just testing this near harvest to farm to fork hypothesis. Right. So that's just getting started. So these other 2 areas we are also very interested in.
But it's the same compounds, right? It's still Phytalix.
It's the same compound, but I should say that's a bit different formulation.
Yeah. Okay. All right. We got some good questions in the chat box. Let's go there. And first from Liz Mahood, how do you think your product broke the growth defense trade-off?
Oh, because it's not activating anything, right? So if you think about, if you put that in perspective as vaccine, right? If you get the vaccine, your body does something that is not enough to change the direction of your body. The same goes to the plants. Priming is just changing something in the genome. That's not enough to change the direction, but that's enough to prepare plants for the future infection. So I think that's the difference between defense activation and defense priming. So we are not activating, or we are activating a little, but we are mostly priming the defense response. We're just preparing plants. We're just telling plants that, hey, there's something may come, just be ready.
Right.
But that doesn't have to change the direction they go by their life. If no pathogen comes, they can do what they usually do, and there is no change in growth and their normal physiology. And this, 3 years ago, yeah, this question, I would say this is what I think. But now we have tested this in the field and even gone to 200-fold concentration that we usually use to see if anything bad happens to the plant. So far, we haven't seen any negative impact on the plants. So that validate our hypothesis of the defense priming.
Yeah, so the way I think of it is, these are pheromones, right, from the worms. So they're kind of talking to each other through these compounds. And the plants have evolved to say, oh, hey, I see this compound. That means there's some C. elegans in the soil. They might come eat me soon. So get ready. And then once they actually started getting eaten, then they turn everything on, and then you get the growth defense straight off, I guess.
Yeah. Well said, AJ.
That's how I think of it anyway. I don't know if that's accurate. Awesome work. I was wondering how you would mass produce this product.
So when we started this project in 2013, the Schroeder lab produced milligram for me. Like, hey, Murli, here is 2 milligram, do your experiment. But that was clearly not a viable commercialization approach. Since then, I think we came this far where we have synthetic lab at Langmuir. That's where we have a chemistry team, 3 of them, including our CEO, Jay Farmer, who's a chemist too. And we get a lot of help from Frank Schroeder. Last 5-6 years, we got from a milligram scale to a kilogram scale now. And this is, although this is a natural product, it's chemically synthesized in the lab. So we have scaled up to a multi-kilo scale now. This is, again, via chemical synthetic route.
Yeah, I think that's important that Frank is a chemist. He works with C. elegans, but he is a chemist by trade.
Mm-hmm.
And Dan Klessig was the plant science guy of the pair. Here's another similar question. I assume you've synthesized a compound and can scale up production. Also, is it sprayed or applied in the soil? That's a good question.
So mostly we are focused on spray now because that's where we see the most viable approach. We have also been trying seed treatment, but the focus with seed treatment has been on the soil-borne pathogens. We don't expect that the seed treatment at early stage will provide protection 4 months after with the foliar pathogens. So mostly spray, some seed treatment. We are also considering soil drenching. It's common practice in some crops like potato. They drench the soil with the fungicides. That's something we are also trying this year. So it seemed like it worked in all these mode of action. It's just where the focus is. The foliar is a spray, soil-borne is seed treatment, and also the root drenching.
Makes sense. Why are nematodes synthesizing this molecule? Is there variation in the structure of this compound synthesized by different species of nematodes?
There is a variation in what. So there is and Frank is the right person to say this. There are many different pheromones, right? I think there's particular one, the sclerocytes. It's hundreds of them, right? And the plants pay attention to just this particular one, this particular compound. And we have tested others as well that produced by these nematodes, but they only pay attention to this one. And the logic behind it, when we did the initial analysis of these nematodes, we found this particular compound by far most sclerocyte produced. So that would make sense for plants to pay attention to this one versus something coming tenfold less. So if in a given environment, if the plant's roots are there, this is the first compound they will see. And then other ones there, but they probably see it later. So by the time they see this, they do their thing. So I think that's perhaps the reason why it pays more attention to this particular one, not other sclerocytes. And there is a difference, the profile of these sclerocytes. The plant parasitic nematodes, this particular sclerosite is the most abundant. But if you go to the other one, I think in C. elegans, that the Schroeder lab works most on, this particular one is not the most abundant one. It's somewhere in the low range. So it makes sense why plant would pay more attention to this one, not to the other sclerosites.
These are the dangerous ones. These are they do the root knots. Is that what they're called?
Well, yeah. So the most of the plant parasitic nematodes in those ones, and these ones the most. And I think I missed other question why they produce it, right? I think they are using this for their own communication, and plants are just eavesdropping. Right? I don't think they're producing this so plant can sense it and do what they're supposed to do. I think these guys are communicating among them by producing this molecule, and that's when plants can eavesdrop and sense that these are around, and eventually they're looking for food, and I am their food. So hey, just prepare for ourselves.
Yeah. Makes sense. Here's one I think that's about the seed treatments. The plant surface, or maybe not. Plant surface already has useful or harmful microbes. How does Phytalix interact with them?
That's a good question, and I don't know answer to those. There is one aspect of the project where we're looking for if we treat seeds or plants or even put in the soil, does this changes the profile of microbiome? And early studies suggest that it does not impact or at least the beneficial microbes, right? And all these fungicides out there, the first question is like, if you're applying this plenty on the soil, does it impact the beneficial, the microbes in the soil, or also the beneficial insects in the soil or around there? And I cannot say this for sure if some of the endophytes, good one or bad one, are changed, but initial findings suggest that the treatment does not, or mostly does not impact the microbial composition of soil or seeds or plants. But I think this is a very preliminary finding. We got to do more to understand this better.
Makes sense. If the immune system of plants is more active than defense because of this product, could plant pathogens evolve to better evade the plant immune system?
That's something to find out, right? I think this questions there is something to it, but if you think about this, is we are not putting any extra pressure on the pathogen. We are making plants stronger, just how the natural way plant would defend themselves. We're just making them a little bit stronger. But would that be constantly applying this would change something to the physiology of plants, or would that eventually change to the fungi? I think it's unlikely, but that's something we'll find out maybe 20, 30 years from now. But I don't think I have a clear answer to that. We think it's not, unlike any other product out there, but I cannot say for sure until I see what happen in next many years when we try this on the plants.
The immune systems already exist, right? You're just kind of...
It does, right? It does exist. You're just, making it better. Would that impact their interaction with plant, interaction with microbes or their pathogens? Could be. It's unlikely. I think at this stage, I would say it's unlikely, but I cannot say that's for sure, 100% surety.
Have you tried to mix Phytalix with other defense activators? Are they working in harmony?
That's a good question. That really is a good question. I think there's one part of the conversation, AJ, where if you have this, because this goes through the plants, and there is the incidences where the pathogen becomes too strong in the field, and anything that goes through the plant immune system is not going to be sustainable. So you need to think about mixing this with either other ACR-based product, other biological, or maybe other fungicides. So say we mix this with fungicides that is recommended rate is 100 gram or 200 gram per acre. Can we reduce that? Because we don't need the full efficacy of these fungicides. Can we reduce that to tenfold? And so we can get some protection while the Phytalix do what it's supposed to do. So we are considering all those, and some of the trials are focused on that. see if there is any synergy between Phytalix to biologicals, SAR-based products, or some of the fungicides that's old fungicides that no longer being used. Can we bring that to the life if we mix this with Phytalix? So that's major focus of the R&D, both in the lab and the field of Ascribe.
That makes sense. As long as it can save the farmers some money, give them the same yield or a little more yield and saves them some money.
Yeah.
That makes sense. Has the DNA sequence responsible for the active moiety of this pheromone been identified? And do you know which gene carries it? That's a good question.
Oh, that's getting into the beats of the molecular biology. We know which genes are activated. We also know which promoter or which genes respond to this. We have done some early what you call sequencing, not sequencing, it's basically just looking for which part of the genome is more open than other after treatment. So we know the genes and the promoter of those genes that respond to Phytalix. And this is the work in progress. I think we are, apart from the Ascribe, that's the paper, the research paper that we have been preparing. So we have the list of genes. So I don't want to give that away yet, but we do know what genes and the promoter regions of those genes and what changes happen in those genes that are responsible for active priming of those markers.
In the plant you're talking about? In
the plant, yes.
Does that answer your question, David? By the way I read that question, it sounds like you're asking about the genes in the C. elegans, but I'm not sure. There's lots of new stuff. Yes, okay, I think it answered his question. Does anybody else have any other questions? If not, I have one more I'll throw at you, Murli.
Sure.
Not exactly about the scientific data, but could you tell us a little bit about how you decided to leave academia and go to industry? How did that come into? How did you make that decision?
That's interesting.
Is that a loaded question?
Yeah, that's a good question. And honestly, and before I get into that, I think getting in academics and as an immigrant with the visa and all these challenges, it was quite difficult early on from moving to academics and get fully on board in the Scribe. But I must say that BTI has been very helpful there, right? I mean, every step of the way, the visa and stuff, the Schroeder lab, Frank Schroeder and Dan Plus, they have been extremely helpful. So it was not a quick transition. It was a slow transition. I was a research scientist with these two researchers and published paper on this, but then eventually it was very clear the immediate impact on agriculture of this product. So that's and then while doing that, even before when we were registering the company, talking to farmers, and there's a gap in the pest management. There's all this problem with the current fungicide and stuff. It was very clear to all of us that this may be a good product for the farmers, but it has to go a long way. And I just came along, I came along, and then I enjoyed doing this. I think we still have R&D team. We have 4 people team here at Boyce Thompson, located at Boyce Thompson Institute. We also have a chemistry team at Langmuir. So I'm not away from research. I do a lot of research. I think these people, the team here does a lot of research. But then going out and talking to farmers, and I can tell you, AJ, if you read a paper and it tells you that this is a problem and farmers is facing, and you go talk to farmers, well, like, what problem? That was problem 10 years ago. It's not a problem anymore. And when we went the first time, we talked to the farmers and we were focused on this particular disease. And everybody says, oh, this, but you don't have to worry about this one. We already figured out. And we were, as an Ascribe, basing grant application based on that knowledge we got from the paper or the other researchers. So I go out to the places where, you just go talk to farmers, and then I learned a lot. I enjoy that a lot. And then eventually interaction with industry, the agricultural industry that produces fungicide or the sell fungicides, I think it was all very different. and I think I liked it very much. So I shouldn't say that I transitioned to industry. I think as a team, started this company, and our goal is to commercialize this technology. So I came along, and while doing it, I enjoyed what I did with Professor Schroeder and Professor Klasse, but I am enjoying this a lot because I see this has immediate impact for the agriculture and farmers. All this regenerative agriculture and sustainable agriculture we talked about, I think this product has the value there. So that keeps me up. That keeps me motivated to do this.
That's great. Do you think part of that comes from your background, coming from a farm?
I do, and some of my my colleagues says that you have this natural talent to talk to farmers. You make them so comfortable to talk to you. I don't know if there is any, but I enjoy. I have spoken to a couple of hundred farmers across the country.
Wow.
I have been to the places that places has not seen any immigrant period, and I've been to the places. Mostly it was nice experience, but there were a couple of incidences that was, if I have to take it back, I probably have not gone there.
Yeah, sorry to hear that.
It's amazing, amazing experience.
We got a couple more questions.
Sure.
Do plants have enzymes for the Phytalix biosynthesis pathway?
So there is a paper we published in 2020 where we found that the plant has the pathways that can modify the Phytalix in a different way, shortening the side chain or adding some moiety in there. But whether I don't think plant has the pathway to synthesize it, so that's why we have to. And eventually, and that goes to people ask us, is it natural product, is it organic certifiable? And we say that not at this moment, because even those natural compound, we are synthesizing in the lab. So eventually, if we want to have this organic certification, we may have to find either plants or bacteria or fungi where we can produce it by fermentation or something.
Mm-hmm.
But plant have some machinery, but not the complete machinery to produce this compound.
And what has brought Frank as a chemist to look for the C. elegans pheromone?
Well, that's the question for Frank, I guess.
That's a question for Frank, I think. Yeah.
That really is the question for Frank.
Yeah, but he looks at for metabolites. That's his thing. It's metabolomics. Okay, I think this will be the last question. Is this product currently in commercial use or still at an investigative stage? If the latter, how long until it will be out there?
It's not in commercial use now because we have to go through this EPA approval, all the safety studies and stuff, and you need multiple years of field trial data to actually understand what concentration, what timing, is it the combination and all those things. If everything goes as planned, that 2025 or 2026, that's when it will be available for the farmers.
Very cool. All right, set your calendars. Put a reminder. 2025. To buy some Phytalix.
Yeah, let's hope so.
Yeah. All right. Well, thank you so much, Murli, for telling us all about this. It was a really fun talk.
Yeah,
Really
it was fun.
appreciate it.
I enjoyed it. Oh, thank you, AJ, for having me.
Yeah, it was great. And thank you all out there for joining us and asking some really awesome questions. And please join us for BTI's next Breaking Ground discussion series on Wednesday, November 16th. We'll be joined by Dr. Sheila Ochigboju, who will talk about her work as Executive Director of the Alliance for Science, where she promotes access to scientific innovation as a means of enhancing food security, improving environmental sustainability, and raising the quality of life globally. So she's doing a lot of cool stuff all over the world. So that'll be a really good one. Please don't miss it. And you can go to btiscience.org to register. And I'm going to put a bunch of a bunch of links in the chat. So you can go to btiscience.org. Oh, they didn't all go live for some reason, to register. And you can also read more about BTI's research and Ascribe in many other neat stories in our annual report, which you can find online at btiscience.org/annual report. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to BTI to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. Thanks again, everyone out there, for joining us. Thank you, Murli, for being a great guest today. And I hope everybody has a wonderful day, and please be well.
Thank you. Thank you, everyone.
You're welcome.
Bye.